Good morning/afternoon, everyone. I am delighted to welcome you all today. I'm pleased to introduce our presentation on a very important topic. Today, we will be discussing out-of-specification investigations through a detailed case study. This session will help us understand the process, challenges, and best practices involved in handling OOS situations. We'll cover key aspects to enhance our approach and ensure compliance. So, let's get started and explore this critical area together. Now, let's take a closer look at the key points related to out-of-specification, or OOS, results. First, it's important to understand that OOS applies not only to raw materials, but also to in-process samples and finished products. Essentially, anytime we test a product or material, an OOS result indicates there might be a potential issue with its quality or the testing process itself. When we encounter an OOS result, it's crucial to follow a formal investigation process in line with GMP guidelines. This isn't something to overlook or dismiss. It requires a thorough and scientifically sound evaluation before we decide whether the batch can be released or needs further action. Now, to clarify, an OOS result occurs when test results fall outside the predefined limits or specifications. These are the standards established in official compendia, regulatory filings, or our internal quality standards. So, what exactly is OOS? It stands for out of specification, and it simply means the test result does not meet the set acceptance criteria. This situation is quite common in pharmaceutical quality control testing, but regardless of how often it happens, it always triggers a detailed investigation as mandated by GMP guidelines. In summary, whenever we see an OOS result, it signals a potential problem that must be scientifically evaluated to ensure the integrity of our products and compliance with regulations. Let's take a closer look at this case study overview. Here, we're focusing on a product, paracetamol tablets, 500 milligrams. The test conducted was an assay using HPLC, which is a common and reliable method for analyzing such compounds. The specification limits for this test are set between 95% and 105%, meaning the amount of active ingredient should fall within this range to meet quality standards. Now, the observed result in this case was 92.8%, which is below the lower limit. This is what we call an out-of-specification result, or OOS. It's a critical finding because it indicates that the product didn't meet the required specifications. This brings us to the importance of investigating OOS results thoroughly. First and foremost, such investigations are vital to ensure the product's quality and, ultimately, patient safety. If a batch falls outside the specification limits, it could potentially affect the efficacy or safety of the medication. Additionally, regulatory agencies like the FDA, MHRA, or WHO require rigorous investigation of OOS results to maintain compliance. Failure to do so can lead to serious consequences, including batch rejection or product recalls, which can be costly and damage a company's reputation. Finally, conducting a proper investigation helps us identify the root causes of the deviation. Whether it's a problem in the manufacturing process, raw material issues, or analytical errors, understanding the cause allows us to implement corrective actions effectively. This ensures continuous improvement and helps prevent similar issues in the future. Let's take a closer look at our initial observation. The analyst obtained a result of 92.8%. Now, this is an important figure because it falls below our lower specification limit, which indicates that the result is outside the acceptable range. Despite this, system suitability tests were passed, meaning the instrument was functioning correctly during the analysis. There were no obvious instrument errors detected, which suggests that the equipment was operating normally. As soon as this out-of-specification result was identified, we reported it immediately to QA. This prompt reporting is critical for maintaining the integrity of our process and ensuring that any issues are addressed quickly. Overall, this step helps us to catch deviations early and keep our quality standards intact. Let's go through the key findings from Phase I of our investigation. First, it's important to note that there were no calculation errors identified, which is a good sign that our data handling was accurate. Additionally, the instrument calibration was confirmed to be valid, meaning the equipment was functioning correctly during these tests. The standard preparations also appear to have been correct, so we can rule out issues with our reference standards. However, despite these positive points, we did observe a slight variation in sample dilution. This is something we need to consider carefully, as even small deviations can impact the results. Moving on to the laboratory investigation itself, our next step is to check for any possible analyst errors. That includes reviewing calculation mistakes, dilution errors, or issues with how the samples were prepared. To get to the bottom of this, we'll review raw data, analyze the chromatograms, and examine the instrument logs. These steps will help us determine if any procedural errors occurred or if other factors contributed to the variation we observed. Overall, this thorough review is essential to maintain confidence in our results and to identify any areas where we can improve our process. Let's take a closer look at our retesting strategy. In this case, we performed the retest using the same sample preparation, provided it was still valid. We also used fresh standards and reagents to ensure the accuracy of the test. Despite these efforts, the result was 93.5%, which still falls outside of our acceptable range, so it remains out of specification. Now, moving on to our hypothesis formation. We considered several possible causes for the out-of-spec result. These included potential inconsistencies in sample preparation, weighing errors, degradation of the sample over time, and variability in the instrument's performance. This phase is crucial because understanding these potential causes guides us in pinpointing the root of the issue, ensuring that we address the actual problem rather than just the symptoms. This investigation forms the foundation for improving our processes and preventing similar issues in the future. In the manufacturing findings, we observed that during the granulation stage, there was a slight overdrying, which can impact the quality of the final product. This overdrying raises concerns about the possible loss of the active ingredient, which is critical for ensuring efficacy. Additionally, the blend uniformity was borderline, indicating that the mixing process might need some adjustments to ensure consistent distribution of ingredients. Moving into phase II, which is our full-scale investigation, we're taking a comprehensive approach. We'll review the entire manufacturing process to identify any potential issues. This includes evaluating batch records to see if there are any inconsistencies or deviations. We're also inspecting the equipment cleaning logs to ensure proper hygiene and maintenance, which can influence product quality. Lastly, environmental conditions are being closely examined, as factors like temperature and humidity can significantly affect the manufacturing process and the stability of the product. All these steps are crucial as we aim to understand the root causes and improve our process to meet the desired quality standards. This thorough review aligns with our overall goal of ensuring product consistency and safety. Let's talk about the corrective and preventive actions, or CAPA, that we've implemented to address the issue. First, we adjusted the drying time and temperature to ensure the granulation process stays within optimal parameters. Next, we updated the standard operating procedure, or SOP, specifically for granulation monitoring, to improve consistency and control. We also retrained our operators to ensure everyone is aligned with the new procedures and understands the importance of precise process execution. Additionally, we've increased the number of in-process checks to catch any deviations early on. Now, when we look at the root cause and CAPA, we found that overdrying during granulation was a key factor. This overdrying led to degradation of the active pharmaceutical ingredient, or API, which ultimately caused a lower assay value. Addressing this root cause through these actions is crucial to prevent recurrence and ensure the quality of our product. This example highlights how thorough investigation and targeted corrective measures can effectively resolve process issues and improve overall product quality. Let's take a look at the final decision regarding this batch. The batch was rejected because of an out-of-specification, or OOS, confirmation. This means that the results we obtained did not meet our quality standards, prompting us to reject it to ensure safety and effectiveness. Importantly, we didn't just stop there. We thoroughly documented the investigation into what caused the OOS, and this investigation was reviewed and approved by the relevant quality teams. This step is crucial because it ensures transparency and that all root causes are properly identified. Following the investigation, we implemented a corrective and preventive action, or CAPA. This is a proactive measure to address the root causes and prevent similar issues in future batches. And we didn't wait. This CAPA was put into place before producing the next batch, demonstrating our commitment to continuous improvement and quality assurance. Overall, this process highlights our rigorous approach to quality control, ensuring that every batch meets our standards before it reaches the market. In conclusion, I want to emphasize that deviation and incident management are essential parts of maintaining a strong quality system. Whenever there's an unplanned event, or if someone departs from approved procedures, it's crucial to identify it quickly, document it thoroughly, and investigate to find out the root cause. Taking a systematic approach here is vital because it helps us assess how these deviations might impact product quality, safety, and compliance. By doing so, we can address issues more effectively and prevent them from happening again. Implementing corrective and preventive actions, or CAPA, is a key part of this process. It not only helps us avoid recurrence, but also fuels continuous improvement across our operations. Finally, proper documentation and timely closure of deviations and incidents are more than just regulatory requirements. They are indicators of our commitment to excellence and operational reliability. When we manage these events well, it strengthens our overall system and ensures we're consistently delivering safe, compliant, and high-quality products. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation helpful and insightful. Remember, we're committed to supporting you beyond this session, so don't hesitate to reach out. Thank you once again, and I look forward to connecting with you all soon.